Give me a second. All right. So, hello, my name is Luis Garabito, and today I'll be discussing barrel capsule formation and thermodynamics. Well, first, I'd like to start off by I had the honor this summer to study under Dr. Ruiz, a theoretical physicist, studying the biological system. So we study biophysics. And in particular, I would like to first start by asking, what is a virus and what is a viral capsid? Well, a virus is an agent that affects a host cell and copies itself, which then leads to a particular reaction in the host. And a cell is made out of a capsid and a genome. As you can see in this figure, this outer shell is called the capsid, and inside is the genome. And there are different types of viruses, such as a single-stranded RNA, double-stranded RNA, DNA, and many more types of viruses. But in particular, in our lab, we focus on the cowpea chloric model virus, CCMV, bromic mosaic virus, BMV, and hepatitis B virus. And something that I learned was also that viruses have this particular symmetry to their geometry. Viruses have icosahedral symmetry, meaning that in the sphere-like viruses, they have a 20 face composed of equilateral triangles. And depending on the T number, it dictates the size and the subunits present in the capsid, which I'll explain in this next slide. So we use the Casper and Klung model to describe their symmetry. If you have a T number of 1, that means that the capsid is made out of 60 subunits, 60 proteins. But as you keep increasing in T number, then say, for example, you have T equals 3, then you have 180 proteins. You simply multiply 3 times 60. And as you see, the configurations the symmetry is still the same, because if you have this pentagon here, you can name that a pentamer, but as you increase in size, you also have to add a hexamer, basically a hexagon. So it becomes 1, 2, 3, 4, 5, 6, pentamer 1, 2, 3, 4, 5. We study cowpea chloric model virus, CCMV, but the question is, why do we study it? Well, CCMV is a virus that infects plants, and it causes yellow patterns to appear on the leaves of these plants. And the reason why we study it is simply because there's an extensive amount of data on CCMV, because there have been a plethora of experiments done on it. And it also has a symmetry of a soccer ball, right? As I explained, the symmetry of the capsids, it has the pentamers and the hexamers. And then over here, you have a picture of the CCMV capsid. And the question is, that's cool, but why is it helpful? So what we study is the capsid formation of CCMV or similar viruses, because they can actually help in the development of nanotechnology and medicine. In particular, in nanotechnology, there are these things called nanocarriers. Nanocarriers can help in delivering medicine to particular areas. In this instance, it can also be applied to cancer. And then it can also help in medicine delivery and also development of antiviral drugs. Now I'd like to talk about the thermodynamics of capsid formation. Variables that actually impact the capsid formation would be the binding energies. I'll get to that in a little bit. Stoichiometric ratio, the pH of a solution, and ionic strength of a solution due to sodium chloride or salt. The concentration of free proteins are basically just floating around in solution, and the core particles. Core particles act like a genome. They can either be gold particles or it can be any type of polymer that actually functions as that. The temperature, free energy of the system, and the entropy. So our free energy equation can be written as follows, where this first term describes the entropy of the free proteins, where entropy is basically the disorder of the system, right? So this basically means that this is the disorder that this brings. Here's the entropy of free cores, and this last term that describes the entropy of the capsid size and the binding energy of the capsid size. So basically, it's one term, but you can also see that's a sum. So there can be multiple. Over here this here basically just describes what these particular objects mean. And what I like to explain is that capsids are made out of proteins, and these proteins actually come together through hydrophobic interactions. And as they come together, the binding energies play a very crucial role into how they combine and stay together. Eventually, from free proteins, they'll combine and basically make a large capsid as here. Now, important ideas and equations to keep in mind are the Helmholtz free energy at constant volume and the Gibbs free energy at constant pressure. For this particular research, our free energy equation can be written as beta F is equal to negative ln of Q, where Q is a partition function. A partition function is used in statistical mechanics to basically tell us the state of a system, or tell us, we can use it to find the entropy, energy of a system, or free energy. It can be used in many versatile ways. Once we find the equation, we can minimize the free energy and obtain the law of mass action. Even though the capsid formation is non-equilibrium, we can use it and we can still find that the law of mass action still follows. Now, the law of mass action in general can be written in such forms. So x sub q is equal to x sub c, x sub s over e g plus h times q, where the e, the exponential here, is the critical concentration of proteins, of the proteins in the capsid. And essentially, by minimizing the free energy, we can find the law of mass action. And what the law of mass action really does is basically to relate the density of the capsid to the density of free proteins and the genome, if the genome is present inside the solution, so that it can actually create the capsid. And something that's actually pretty interesting is that in experiments, there has been seen that there can be multiple sizes of capsids present. And that phenomenon is called polymorphism. So the thermodynamic equations of polymorphism of free energy can be written as follows. It's similar to the one I showed you before, where you take into account the entropy of free proteins, entropy of the free core, there's a genome, and also the entropy and energy associated, binding energy associated with the different capsid sizes. And in the absence of a genome, because that's what they also found, is that there are the situations where the capsid will form in the absence of a genome, or there would be no genome inside the capsid. But in this instance, you wouldn't take into account the entropy of the free cores or the particles. And in this figure, we actually have the description of polymorphism. As we see here, as the pH is lower than 6 and the ionic strength is lower than around 0.4, you see that there's coexistence of multiple capsid sizes. But as you increase, you have the dissociation of free dimer, dissociation of the capsids, and it comes to dimers, which basically are molecules with a pair. And as you go up in ionic strength and lower pH, you have the T equal 3 capsids. So the final discussion, what I'd like to basically conclude this thermodynamic explanation is that thermodynamics looks at capsid formation as a chemical process that can go back and forth, right? And that the law of mass action can be used, well, actually explains this process pretty well. And that by using Helmholtz free energy, we can derive an equation that helps describe the entropy and energy associated with capsid formation. And eventually, by laws of physics, it'll eventually reach an equilibrium. The next step in my research would be understanding this scenario. Yeah. So it was found that under certain conditions, CCMV or Cowpea-Clark model virus would dissociate in a solution and recombine to different T number capsids in the absence of a genome. So we look at this figure here. If you have T equals 3 protein capsids, a capsid at pH 7, if you were to increase the pH to 7.5, the proteins would dissociate and then recombine into T equals 1 in the presence of elastine, a polymer. And then if you were to decrease the pH back down to 5, well then that capsid T equals 1 would dissociate and recombine to T equals 3. And what we're interested in is describing why. Why is it doing this? And over here, you can see in this figure that initially there is a 0 concentration of T equals 1 capsids. And over here, there's a large concentration of T equals 3. But as time progresses, T equals 1 increases and the number of capsids of T equals 3 decreases. And a way to actually explain that formation is a theory that Dr. Zandi has been working on, just to explain through classical nucleation theory, or CNT. And classical nucleation theory is basically the most common theoretical model used to quantitatively study the kinetics. Kinetics is basically the movement of particles and description of that movement of nucleation, right? So, and it actually describes nucleation. Nucleation is the first step of spontaneous formation of a new thermodynamic phase or a new structure, starting from a state of instability. I want you to think of a supersaturated solution, and if you were to put a stick on there, eventually all those particles will basically start to form around the stick. So think rock candy. That's what these capsule formations are like. They're basically coming together and forming a new type of phase, thermodynamic phase. And that's what my next step would be, to learn more about that and explain the kinetics of capsule formation. And lastly, I'd like to acknowledge my faculty mentor, Ruiz, my group members, Mark Nugent, Tony Montalvo, Alreza Ramzani, and Celine Dora. Our collaborators, Paul van der Schoot, Suzanne Timmermans, and programs CNAS, RISE, and Summer-Based Research, and all the peer mentors, and Mr. Nee, and also our funding NSF. And these are my references. Thank you very much.